Okay, welcome everybody to our Monday HNRCA seminar. And today I think is going to be a really exciting seminar. So our speaker today is Tim Rhodes. Tim obtained his Ph.D. at the Oregon State University in biochemistry and biophysics. And from there, he went on to do his postdoctoral fellowship under the mentorship of Rosalind Anderson at the University of Wisconsin in Madison. Tim has some very, very impressive publications, particularly in the area of calorie restriction. His most recent one being a perspective on metabolism and calorie restriction that was published in Science as the perspective for the series of publications that were emanated from the doubly labeled water team on energy requirements across the lifespan. Tim has also published in very prominent journals, including a paper in Cell Metabolism, in which they looked at the effects of calorie restriction on hepatic RNA processing mechanisms in research monkeys. Tim is well-funded for someone at his stage of career. He currently has an NIH R03 looking at the molecular consequences of protein regulation on amyloid plaque burden. I could go on at great lengths of all Tim's accomplishments, but I think you're all here to hear his exciting seminar today, which is going to be really on the molecular mechanisms underlying the benefits of calorie restrictions. And as he says, his talk's going to be more fun than a barrel of monkeys. So with that, I'm going to hand it over to you, Tim. We're all looking forward to your talk. Thank you.
Thank you, Sarah, very much for the kind introduction and for the invitation to talk. As Sarah mentioned, I'm going to talk a little bit about caloric restriction and mechanisms of delayed aging and what we've been trying to learn and what we've been able to learn using large-scale molecular profiling techniques and studies from the long-running non-human primates aging and CR study that's been ongoing at the University of Wisconsin for quite some time. Just briefly, since I'm sure I don't need to introduce caloric restriction to this audience, caloric restriction is a reduction in the daily caloric intake, and back in the 30s, Clive McKay found that if you fed rats less, they lived longer. And so what I have on the slide here is some somewhat updated rodent data from Rick Weindrich's lab in the 80s in mice, showing the body weight and percent survival on different levels of caloric restriction. And the interesting thing about this is you get sort of a proportional, almost a dose response to the level of restriction, ad libitum 25%, 55%. And if you plot lifespan versus caloric intake, you get what amounts to almost a linear relationship between the two, at least in rodents. And so when I look at that, and Roz, my boss, looks at this, it sort of screams metabolism to us. The fact that daily caloric intake that you would derive energy from is ultimately linear related with lifespan. And so we look at this through the lens of metabolism in terms of how we think CR works. Now, CR, as I mentioned, was first sort of described in the 30s, and it's been since tested in a lot of different model organisms. And it seems to work pretty universally, with obviously a few caveats. But for a long time, it was sort of an open question about how well it would work in primates, specifically humans. And so in the late 80s, 2 studies of CR and aging were started at the University of Wisconsin and the NIH. And so these are using the rhesus macaque animal model. So these animals, this is an old world monkey. They live about 26 years or so in captivity. And the University of Wisconsin study started with 76 animals, so 46 males, 30 females, and was designed to test a 30% restriction that started when they were roughly young adults, so full stature, fully developed young adults. And I won't compare too much to the NIA study on rhesus monkeys, except to say that there are some people that think there's kind of a controversy between the two studies, and we don't think so. The combined data analysis has shown that CR works in non-human primates. And when we look at the Wisconsin study in particular, we think of this as a successful implementation of CR. So as of earlier this year, actually, the last monkey has died, and so we have complete survival curves. And if you look at all-cause mortality, but also age-related mortality, you can see that there's a statistically significant benefit in the caloric restricted animals, almost twofold in all-cause mortality and fourfold in age-related mortality. So it appears to have worked in these non-human primates, and so that's a great thing as far as the translatability of the mechanisms. And so building off of this, what we have been trying to do over the last few years is to sort of try and figure out at the molecular level, what is it about CR and what CR is doing that is resulting in this extended longevity. And so one of the things I like to throw up, I know this slide is a little bit busy, but coming from a mouse background and being used to sort of designing studies in mice, I think the mechanics and the design of this study is sort of interesting and a tad overwhelming, actually, because it's a study that has outlasted the careers of its originating principal investigators. And so this has been ongoing for about 30 years. And so what you have here, this is all of the necropsy collections of tissues. Some of the animals for several tissues had biopsy collected very early on. And I'm going to talk about one story related to those. There was also some biopsies collected of muscle across all of these different groups. And these animals have also been sampled in a lot of different ways by what we call biometrics, so body weights, body composition, blood measures. We have functional data, physical activity, metabolic chamber, and doubly labeled water from different time points, all sort of in service of trying to maximize what we can learn from this study. So there's a humongous amount of data that has been collected on these animals so far, and we have a very large amount of samples still in the freezer waiting to be looked at. So I obviously can't talk about all of it today. So I'm going to focus on sort of 3 short stories of CR that I've been involved with. And they all differ in terms of the tissue and sort of the timing that we're looking at. So the first one, and this was published in Cell Metabolism a couple of years ago, is the hepatic short-term response to CR. So these are using those biopsies from enrollment and 2 years into the study from male monkeys. The second story is using biopsy tissues from skeletal muscle at a couple of different time points, and we have several different age groups in here, and as well as both males and females in the late middle age group, so we have a chance to look at sort of sex dimorphism. And then the final story, and part of this was published last year in Cell Systems, and then the last study is going to be on adipose tissue and from necropsy samples, so lifelong CR, and this one hasn't been published yet. So the first story, the liver response to short-term CR, what we did was what I call kitchen sink molecular profiling. Every conceivable molecule type that we could assess, we did. So that includes RNA-seq, proteomics, post-translational modifications, acetylation in particular, lipidomics, and metabolomics. So all told, 22,000 molecules that we profiled across all of these different monkeys. And we have, some of these data sets have different numbers of animals in them, but we have 8 animals for which all of these profiles were collected, and that's largely what I'm going to focus on. So starting with the transcript data set, which is the sort of the richest data set that we have, pathway analysis, what we see is upregulation of metabolism. branched chain amino acids, oxidative phosphorylation, TCA cycle, largely a metabolic signature across these upregulated pathways. And then in terms of what was downregulated, we see a lot of inflammation and immune-related pathways, cytokines, chemokines, cytotoxicity. And so all of this pretty much matches what we would expect from other organisms and other data sets. CR engages metabolism, and it has a tendency to down-regulate the immune and inflammation pathways. So this was sort of gratifying to see that those mechanisms are indeed translatable. The one piece of this that was sort of interesting to us was the Rna Transport and Spliceosome, and there are a couple of other pathways that aren't labeled that are sort of RNA related. Around the time we were trying to publish this, Will Maer's group at Harvard published the requirement for splicing factor 1 to get the benefits of dietary restriction in nematodes. And that was sort of a codification of some anecdotal observations about the involvement of RNA in the mechanisms of CR from previous work. And so we wanted to dig into this a little bit more and see if we could figure out what was happening, what exactly were the details. And so as a first pass, what we did was use a tool called DEC-seq to look at our short read RNA-seq data. And what this does is it just tracks the sequencing reads through exon junctions and looks for changes in the inclusion and exclusion of exons. And when we do that, what we found was that in the control animals, this really isn't happening. And that's perhaps to be expected. In the CR animals, however, we had 387 genes for which we had at least one exon that was being differentially used as a result of the diet. And so we think this is another piece of evidence that suggests that RNA processing in some form is an important part of CR's mechanisms. And this is happening in a primate. So we thought that this was kind of an important thing. And I'm going to bring this back when I get towards the end of the story and start trying to put a bunch of these data sets together. But we also see a lot of these same themes in the other molecular types. So if we
Tim, are you having audio problems?
everyone hear me now?
Yeah, you're back.
Okay, I don't know why those just decided to turn off, but anyway, sorry about that. So when we look at the proteome data, we see a lot of the same pathways engaged. So ribosome, spliceosome, proteasome, peroxisome, metabolism, homeostasis, potentially RNA processing. And the interesting thing here is perhaps that these pathways aren't really going in the same direction necessarily. Peroxisome in particular in the transcripts seemed to be upregulated, whereas here we're seeing a trend towards downregulation. And this is sort of interesting. It's also well established at this point, I think, that transcript and proteins don't always accord. And we also are not necessarily capturing the exact same factors that are present in the transcript data set. The proteomics data set is quite a bit smaller. So we don't necessarily know exactly what this means, but it was encouraging to us that we were still seeing the engagement of these same pathways. And the other thing I'll note is that most of these individual factors are not statistically significant. And this is something I'm gonna harp on in the talk, because with the heterogeneity of the animals and sort of the variance that it goes into analyzing aging, traditional statistical testing often doesn't give us much to work with. So a lot of what I'm going to describe is sort of our attempts to put data together in different ways so that we can try and learn something independent of the traditional statistical tests. When we look at acetylation, this was another one where the pattern in the data set was kind of interesting. By and large, the acetylation was upregulated in response to CR. And this was a bit surprising, I think, to some people. The somewhat traditional view of acetylation was that it was inhibitory and that increases in acetylation were not necessarily going to be positive for enzymatic activity. And I should note here that I'm talking about non-histone acetylation. So acetylation and its consequences, I think, for histone-based marks are a bit more well worked out than the individual consequences of acetylation in a non-histone context. And that's the bulk of the acetylation that we were observing in these samples. And actually, the vast majority of it was occurring in the mitochondria. And this sort of makes sense, given that CR tends to push metabolism towards lipids. That would have a tendency, I think, to increase acetyl-CoA, and that is where this acetylation is probably coming from. We have not individually tested any of this yet, but that sort of regimen, I think, kind of makes sense to me. And so you can see across the mitochondrial marks, there's lots of metabolism enzymes and lots of upregulated acetylation marks, which, like I said, I think is coming from the availability of acetyl-CoA and is telling us something about the levels of acetyl-CoA in the mitochondria. Speaking of lipids, in our lipidomics data set, we have about 350 lipids that we profiled. Some of them were individually statistically significant, but it's sort of hard to know what the consequences of an individual lipid changing are. And so we tried to look for some larger scale trends across the lipids. And there were two that we thought were sort of noteworthy. The first was that across the phospholipids, we saw sort of this uptick in odd chain fatty acids, which was a little bit interesting. There's a couple of different sources for odd chains, but they're not common. One would be methylation of fatty acid chains, and then the other is that they're potentially coming from microbiome, bacteria derived. Both of those would look similar to a mass spec and show up as an odd chain. So this was sort of an interesting feature. The second piece is in the triglycerides. We noticed sort of a bias for unsaturation. So triglycerides with an increasing number of double bonds tended to be upregulated across the CR animals, which is also sort of interesting. And we don't really know what that means. There's a lot of possible, double bonds are sites of potential lipid peroxidation, which goes up with age, but it's not really clear what this is doing mechanistically, but an interesting observation. And I'm not going to show the metabolite data. There wasn't a lot changing in the individual metabolites. We saw some slight changes in the branch chain amino acids as a whole, which dovetails with what we saw in the transcript data, but nothing really substantial. And so the next thing we try to do is to try and kind of put some of this data together and see if we can't get some insight from looking at combined sets of data. So there's a couple of strategies we used for this. The first one is depicted here, and this is a giant correlation matrix. So every single biological molecule correlated against every other biological molecule that we measured. So it's about 450 to 500 million individual pairwise comparisons. And then this has been hierarchically clustered to try and look at where we have patterns of similarity. And so we end up with this pattern that kind of shows sort of these 7 mega clusters. And I'll note that if you impose this hierarchically clustered order on just the control animals, for example, the pattern completely disappears. There's no, so this appears to be the result of the CR diet. And if you look in these individual clusters, there's basically lots of metabolism happening in terms of the pathways that are engaged in all of these clusters, including some RNA processing in multiple different data types. And we haven't yet dug into if there's specific things that are only in one of these clusters versus another. But we have metabolism sort of engaged across a lot of these clusters. And that includes big changes to a lot of the fatty acids as well. So that was kind of a huge avalanche of data. And as I mentioned, we see a lot of the common themes, metabolism with an exclamation point. You'll get tired of me saying that, I imagine. Also downregulation of growth, immune and inflammation, which is pretty consistent across most of this data. Lipid and metabolite changes, the somewhat unique observations, upregulation of acetylation, and this RNA processing feature. And so that's a lot of very descriptive, what is happening in response to CR. And so we wanted to try and see if we couldn't get a little bit more mechanistic in terms of this. And so the way we do that, as I mentioned, statistical testing in the traditional t-test type of way often is not our friend with these monkeys. And so we used an alternative approach. It's kind of a machine learning type approach where we were generating random forest decision trees. And all of this is you're taking all of the molecules and randomly assessing them to see how well they segregate the animals in terms of their diet groups. And you hold some of the data out as a test set. And then you do this thousands upon thousands upon thousands of times, and then you average them all together and assess each individual molecule for how much it impacts the accuracy of your decision tree. And so this is an example of the output we get from something like that. This is we did each of the data types individually. So this is from the proteomics data. And so you can see we have this list of factors, and what is plotted is how much they affect the accuracy of that decision tree in terms of discriminating between control and CR. And I always like to point out that the top protein is quaking, which is an RNA binding protein, which I don't think is a coincidence. And so taking these lists of molecules from protein, transcript, and acetyl protein, I tried putting them into the string database to see if there's any sort of a protein-protein interaction network. And for each individual data set, that really didn't produce much. But if I throw all three data sets in together, all of a sudden we got this very, very tightly integrated protein-protein interaction network. And it's got lots more edges than you would expect by random chance. And what you can kind of see is that in a lot of cases, there's sort of these little mini clusters, and their only connection to other clusters is a molecule of a different data type. So this is something we were only going to be able to see when we included all of the different types of data that we had. And the overwhelming majority of this network is really focused on metabolism. And so then the other strategy we like to use is to try and layer data together. And so the question I asked was basically, across this network that we have here, how many of
Tim, your audio is still not working.
There we go.
Yeah.
Should be back now. How many of these factors are, of these 387 factors, are present across this data set? And it turns out to be an overwhelming majority of them. So across this integrated network, more than half of this network is subject to that RNA processing regulation that I described at the beginning. And so we felt like this was a little bit more of a, not a direct mechanistic connection, but at least putting data together that suggests a possible mechanism. And that's that RNA processing is part of sort of setting this metabolic program in response to CR. So as I mentioned, metabolic reprogramming in multiple molecular layers, a few new observations that came out of this data, some interesting ones with regards to fatty acids, increased acetylation, and RNA processing that was sort of observable across this whole metabolic network. So all of these things are things that we're actively still investigating, along with some other questions like, this is short-term CR from very early on, so it's not yet clear how much of this persists and how connected it is directly with the longevity effects that we see. So the next little vignette that I'm going to talk about is the skeletal muscle response to CR. And this is the molecular characterization of biopsy tissues from a couple of different groups of animals. So we have very old males, 27 years of age, about the median lifespan. And then we have some late middle-aged males and females who are about 22, 23 years of age. And so we were able to do the same sort of high throughput molecular characterization, although in this case it's microarray and microRNA. And I will try to say those two very, very clearly because they sound very similar. But then we also have cryosection tissue from these animals, and so we can assess some immunohistological measures as well. And so when we look at the very old males, this is again a pathway analysis from microarray data, and we see sort of very similar patterns as we saw in the liver. So homeostatic and energy metabolism pathways are upregulated. And immune and inflammation related pathways are kind of downregulated. And so those appear to be tissue agnostic mechanisms of CR, and they're engaged in multiple different contexts. In these guys, we can kind of look a little bit more closely at what that might mean in terms of mitochondrial energy metabolism is upregulated. And so this is looking at cytochrome C oxidase staining. And this is sort of a 3D reconstruction of the intensity of an individual fiber from a control animal and a CR. And yes, I picked this one because it looks like the shape of Wisconsin. But one of the things that has been observed by Roz's lab in the past is that there's a stark difference in terms of the activity levels of cytochrome C oxidase, which is complex 4, between the perimeter and the mid fiber. And so it's much more strong on the perimeter and lower in the mid fiber. And this is a difference that goes away with age. And so the difference you're seeing here in the CR animals being much more intense, this is a more youthful phenotype for the muscle fibers. And so this is quantified here on the right. You can see that the gap is still there in the control animals, but it's larger in the CR animals. And we think this is related to the fact that their muscle overall is just in better shape than the control animals energetically. And this sort of extends to the overall metabolic environment in the cell. So this is looking at 2-photon data, fluorescence lifetime imaging microscopy. Basically, you are exciting the endogenous fluorophores, NADH and FAD, and looking at the decay of the timing of the decay of the fluorescence. And this says something about the overall environment in which the fluorophore exists. So there's an equation that can be used to fit the data, and contributing coefficients include things that indicate the level of bound and unbound cofactor, and then just what the overall environment, how it is interacting with the metabolic cofactor. And what you see here is that the control animals have much longer fluorescent lifetimes. So this is looking at NADH in particular, and the CRs are much, much quicker lifetimes. And this is consistent with a more youthful phenotype. So this is the quantification. This is 2 of the coefficients. One is the mean lifetime in the equation, and this A1 is sort of roughly indicative of percent bound and unbound. So the metabolic environment is very, very different between the 2 diets, and the CR animals look more like young animals than the controls do. This extends to the lipid metabolism as well. So this is using oil red O to stain the muscle tissue, and oil red O stains for neutral lipids. And what you can see in the controls is that the lipid droplets, which are heavily used for energy in muscle are big, and there are fewer of them. When we look at the CRs, what you see is all these little, they almost don't look red anymore, but they're little tiny droplets, and they're far more numerous. So this is also consistent with a slightly younger phenotype and the idea that these lipid droplets in the CR animals are accessed much more frequently. And that leads to them being smaller and more numerous than in the control animals. So as I mentioned, we kind of like to try and put data together where we can to try and learn something about the overall picture. And so we have a bunch of systemic data for these animals, and this is using a technique called multiple factor analysis, which you can think of like basically a generalized principal component analysis, except this allows you to incorporate multiple different data types. And it's a way to sort of look at how the variance structure across the data looks. And so if stuff falls in a similar area on this variance map, it's probably varying in a related manner. So it's perhaps not surprising that body weight and total lean mass are varying very similar to percent abdominal fat and percent fat. The connection that was not as immediately obvious was the one down here where you have upper leg lean percent of peak. So these animals were assessed every 6 months for body composition. And so we know at what age they achieved their peak muscle mass. And we like to use that as sort of a reference point and report the percentage of their peak that they are at. Because the CR animals maintain their peak muscle mass much better than the control animals. The interesting thing here is that seems to be varying very similarly with SI, which is an index of insulin sensitivity. At first glance, this was not immediately obvious, but it perhaps makes sense because skeletal muscle mass is so important for glucoregulation. The other side of this is looking at where these animals fall in the variance space. And so to talk about that, what I'll note is that for most of these individual measures, we don't have statistical significance in terms of the difference between control and CR. When you consider all of these together, though, this is where you get a pretty good separation between control and CR animals. You can see that the control animals are kind of spread out, and the CR animals are a little bit more tightly grouped. This control animal thinks it's a CR. It's really not. If it perfectly separated, I wouldn't believe it. So I think it's good that there's one sort of outlier. But when we consider all of this data together, we get a pretty decent separation, which was nice to see. And so then one of the other things we looked at was microRNAs. So these are small 22 nucleotide RNAs that regulate gene expression via RNA interference pathways. And so one of the things about this data set is that no individual microRNA was statistically different between control and CR. Story of my life with these data. But you can look at microRNAs as groups in a couple of ways. So one is they can be grouped by family, which is basically sequence similarity. They hit similar targets because they have similar sequences. And you can also look at their location in the genome. So you'll have clusters of microRNA genes that can all be transcribed. Can all be transcribed in a line by POL2. And when we do that, what we see is that there's this one chromosome 7 locus that is downregulated as a group in response to CR. And this is a locus that has been previously linked to things like diabetes and cancer, to age-related chronic diseases. we thought that this was interesting and is very much of interest in terms of exploring sort of coordinate regulation of these microRNAs and how they affect both tissue resident and circulating microRNAs, circulating physiology, excuse me. Okay, so now I'm switching over to looking at the late middle-aged animals, males and females. And this is, again, the microarray transcriptional data. And what you can see when you look at these is that the pattern is not really the same in terms of what's statistically significant. And in fact, the individual factors are not at all the same. There's only 23 in common. And when we compare the old males, there's also very, very little overlap in terms of the individual factors that are engaged. If you look at sort of a network perspective, and both of these males and female transcriptional data sets, if you put them into string, they do produce a tightly integrated network. And there are some commonalities, but in general, it seems that the females are sort of hitting energy metabolism, some of the more traditional response to CR things that we saw in other contexts, whereas the males are much more on the homeostasis, catabolism, and repair pathways. And I don't have time to go through all of this data, but this sort of dovetails with a bunch of the other data we have from these animals that suggests they're not at the same point in terms of their development of sarcopenia. The females are still a little bit earlier. They don't have frank sarcopenia just yet, whereas the males do. And you can kind of see this if you look at their estimated skeletal mass as a fraction of their body weights. So in the females, the control in CRs, there's good separation, but the CR animals are still, pretty widely spread. And you've got some of them that still have a reasonable fraction of their body weight as skeletal muscle mass, whereas the males, notwithstanding this one, there's a very stark difference between them. So like I said, we think that they're kind of just not at the same point in terms of the condition of their muscle tissue. And we see differences when we look at these microRNAs again. And in fact, in the females, somewhat surprisingly, we see a very similar pattern with this chromosome 7 locus. In this case, I don't think it's quite statistically significant, but the trend appears to still be there. Whereas in the males, it's almost going in the opposite direction. And we really don't know what to make of this, actually. It was sort of very surprising that it was doing this. I would have expected the opposite. And so we're still trying to explore what's happening here. So quick summary, more metabolism across these data. And potential regulation by microRNAs, which is something that we're very interested in. It's also very interesting to be able to see sex dimorphism both at the physiological and at the molecular levels. And this is something that I think there's an increasing awareness of, and we don't have enough data on females in a lot of cases. And so this is something that's going to be explored much, much more going forward. Okay, so then the final little story is going to be looking at adipose tissue response to CR. And so this is transcriptional characterization of 2 adipose depots, visceral and subcutaneous. And these samples were collected at necropsy from 8 age-matched animals. So this is going to be slightly different than the other 2 stories because in all of those other cases, the animals were matched basically for how long they had been on the study. And they were all sort of slightly different ages. These guys were age matched, so they're not necessarily on the study for the same length of time. And so the first thing we wanted to do was just kind of compare the transcriptional landscape of the depot. So this is just looking at the control animals. And what I have plotted here is this is just a rank order plot of all of the transcripts. So I've basically ranked them to most highly expressed to most lowly expressed. And then they're also based on filtering we identified some transcripts in each depot that are essentially not expressed. And what we wanted to know is, are there a lot of these that are not expressed in one depot and are expressed in the other depot? And are they changing dramatically between the two depots? And so that's what the line connections are. And so you can see there are some genes that go from not expressed to sort of middle of the pack in the other depot. And so this was potentially very interesting. There wasn't a strong biological theme across those factors. And it's also amounting to only about 500 or 600 total transcripts out of the 14 or so thousand that we quantified. So the takeaway story is really that these depots are extremely similar in terms of where their transcripts rank in expression. Their transcriptional profiles they're very strongly saying, we are adipose tissue. And it was hard to find strong differences between the two depots in the control animals. This is interesting because we did see a fairly substantial difference in their response to caloric restriction. And the subcutaneous transcriptome is responding much, much more strongly than visceral. So you can see in these somewhat messy MA plots that there's far more factors engaged in subcutaneous in response to CR than there is in visceral. And here again, we kind of see that there's not great overlap in terms of the individual factors engaged. So only 18 out of several hundred transcripts are in common responding to CR between the two depots. Also, like with the muscle tissue, although I didn't specifically explore this, I suspect that the biology in the muscle is probably, there's more overlap there than it might appear. And that was true in the adipose tissue. So if you look at a pathway analysis, there's far more pathways in common than there were individual factors. So some of this is down to the stochasticity of what is and is not statistically significant. But it appears that the very similar biological response from a pathway standpoint. And so if you look at these pathways and sort of plot them in terms of their relative expression, you can see across this axis of stuff that's moving in the same direction in both depots, ribosome, spliceosome, RNA degradation. So those are some of my favorite pathways lately. And then interestingly enough, both of them seem to show a signature of down regulation of energy and amino acid metabolism. This is different than what we saw in liver, and something we're still sort of wondering what's happening here. But then there are some stuff that is moving differently in the two depots. It's neurodegenerative pathways, which tend to be electron transport chain. That's the predominant signature that it falls under those pathways. Upregulated in visceral, but downregulated in subcutaneous. So there are some clear differences between the two depots. And this might be down to their cellular composition. So we worked with Devere Ran at UCSF and Roman Camarda at the Buck to deconvolute the RNA-seq data. And what this involves is taking transcriptional profiles of pure cell populations and then mathematically sort of assessing how much of a contribution they are making to your transcript data in question. And so when we did this on the adipose tissue, we see that there are some differences between the depots and in how they respond to CR. So these are relative contributions from adipocytes, endothelial cells, macrophages, and monocytes. And you can see the pattern in response to CR is sort of similar between the two depots. So from here to here for subcutaneous and here to here for visceral. But then in like the monocytes, for example, the change is not the same for the two depots, and they don't necessarily always start at the same point either. So this might contribute to some of the differences that we were seeing in the response. One of the other things I wanted to look at, as I've mentioned, I'm very interested in RNA processing and what is occurring there. And so one of the first ways we tried to do this was just looking at annotated isoforms. So transcripts have isoforms, and although the monkey transcriptome, it's not as well annotated as not as well annotated as it is in mice, but there's about 2,200 of the transcripts that have known isoforms that we can look at. So it's, I think, 12 to 14% of the total transcriptome that we saw. And so if we just sort of layer this over the top of the data that is the transcripts that are statistically different between the two depots, they're sort of scattered across in both depots, and they're both still around 12 to 14% of the genes. So there doesn't appear to be a particular enrichment for them that would be indicative of any sort of an RNA processing phenomenon. But we did also look at DEXseq, that same tool to sort of track exon usage changes. So what I have plotted here are just subcutaneous, so these are exons and corresponding genes, and just a nominal p-value cutoff. It seems like it's going crazy, but there's like hundreds of thousands of exons and hundreds of thousands of statistical tests. So when we control for a false discovery rate, that knocks those numbers down quite a lot. And we see that in subcutaneous, there's about 129 genes, and it's really not happening in the visceral. So this is still consistent with the idea that CR engages RNA processing, and so it's perhaps makes sense that it's occurring in subcutaneous adipose, and it's really just not in visceral, which was a much weaker responder to caloric restriction transcriptionally in the first place. But this is not anywhere close to the magnitude that we saw in the liver story. So the quick summary of this is that the depots are very, very similar transcriptionally at a baseline level, but they do not respond the same to CR. And this difference might be partially driven by differences in cellular composition. And finally, we see that this RNA processing, while it seems to be occurring, it's not as clear of a signature, and we haven't been able to explore it as being as tightly integrated as we did in the liver story yet. So the overall summary, like I said, you get tired of me saying it: metabolism, metabolism, metabolism. That is what CR does. It sort of metabolically reprograms, and we think that is how we get to extended longevity. Obviously, there are some variations in terms of the tissue and timing specificity and what gets engaged in certain contexts versus others. And the other thing I always like to mention is this sort of data integration. It's especially important for these monkey studies, but you don't get the whole picture with just one data type. And so we think we've managed to get a lot more insight from these animals by putting data together in different ways. And that's always the strategy that we like to pursue. So with that, obviously this monkey study has a very long history and is a huge undertaking, even in just what we do in the current day. And so I have a lot of people to thank. Roz Anderson, obviously, for being sort of the leader of all of this and being a fantastic boss. A lot of people in the Anderson lab, both past and present. I do want to mention Grace Gustafson and Joe Clark in particular. So any of the data analysis that I didn't specifically perform myself that I showed was performed by one of them. And obviously we have a lot of amazing collaborators that help us analyze these data in a lot of different ways. But I also want to specifically mention Ricky Coleman, who's she's an associate professor here and also a scientist at the Wisconsin National Primate Research Center. And she's been the co-leader on this monkey study with Roz for a great number of years now. And none of the monkey stuff really would happen without her at all. So very grateful to Ricky for her involvement. And also funding from the NIH, the NIA, and the UW-Madison Diabetes Center we're also physically located in a VA hospital. So we get support from the Department of Veterans Affairs, which we're also very grateful for. And with that, that's all I have. Thank you very much for listening. Oh, Sarah, I think
Sarah, you're on mute.
Thank you, Tim.
And I apologize for all of the interruptions with my headset. I think it's meant to be a Bluetooth, and it thinks it's not connected, so it kept shutting itself off to save battery. So I do apologize for that. Hopefully it wasn't too much of an interruption.
So Tim, thank you for a wonderful seminar, and I am very impressed by your patience with your headset. You handled it beautifully. And I was remiss in my introduction of you. It's my understanding that 2 weeks ago, Tim actually received a promotion at his current institution. He is now a scientist 2 at the University of Wisconsin in the Division of Geriatrics, Department of Medicine. So congratulations, Tim, on your promotion. So with that, I'm going to go over to the questions. I'm just going to read them out and let you go. We're going to just everybody, there's a lot of questions there. So if you asked 5, you only get to, and we're only going to list 1. So the first question was, in the data set, you mentioned there was an interesting observation about the difference in the PUFA. Was there a difference in the absolute amount of essential fatty acid in the diets of the 2 groups of monkeys, or were essential nutrient levels similar between the CR and control animals?
All of the macronutrient profiles were very, very similar, or the same actually. And the only thing that's different is that the CR monkeys, for some essential amino acids and vitamins, they get a slight enrichment in that to compensate for the reduction in the amount of the diet that they're getting. But other than that, nutritionally, the diets are identical. So there should be no difference in terms of the PUFA levels or anything like that.
Okay, now the next question is 5 components. So I'm going to take liberties here and only ask you 2 parts of it. First of all, congratulations on an interesting talk. Was there any evaluation of restriction of protein, methionine, fat versus carbohydrate?
Not that I'm aware of, or at least not that I have touched in terms of data. I'll note that was one of the big differences between the UW and NIA study, and it's something that Rafa DeCabo at NIA has been exploring. I think he published in Cell Metabolism either a year or 2 years ago, where they put a whole bunch of mice on diet composition that mimicked the 2 monkey studies, and actually weren't able to find any difference in terms of the overall outcomes on CR. So that hasn't been explored specifically in the monkeys, but we don't think there's going to be a big difference as a result of that.
So another question from the same person, and then if we have time, we can go back. Are there any proteolytic pathways actually activated, and by which diet?
So there are definitely proteostasis pathways that was most prominent in the skeletal muscle tissue. That's perhaps not surprising. And I believe the proteasome was also enriched in the transcript and protein data sets in the liver as a result of CR. The exact downstream consequences of that are not totally clear yet. We haven't investigated them specifically. But yes, some of those proteostasis pathways are engaged by CR.
Great. Next question. In terms of adipose tissue response to CR, did you see any differences between sexes?
We don't have that data yet. So the data that I presented was only from males. And actually, I think we just got back a huge amount of sequencing that would cover both males and females. But so far we haven't looked yet. I suspect there probably will be some good differences there, but we haven't looked at the data yet.
We're going to have you talk about this later in the future.
Yeah.
Next question, again, congratulations on your talk. From a 20,000 foot view level, were there any interesting results that appeared counterintuitive or unexpected when compared to CR data from worms, flies, or mice?
I think that we had a lot of things that maybe haven't been specifically explored in some of the other organisms. I don't know that I would say that there was anything from a high level view that was contrary to anything that we've seen before. We haven't gone in and specifically compared all of the different pathways that were engaged at these different levels yet. It's actually something I proposed to Roz, that we do sort of we have enough transcriptional data from all of these animals that we could do our own sort of meta-analysis and then compare that to other organisms. But I wouldn't say that we've really looked at this yet in any great detail. So but it's something I think is probably worth doing.
Great. Another question is, again, congratulations on your talk. For the genes showing differential exon usage in the first part of your talk, did you look at epigenetic and regulatory features? Any overlap with aging-related changes such as DNA methylation, chromatin accessibility that corroborate?
So in terms of a pathway analysis from those genes that had exon changes, we didn't observe anything like that. And I would love to look at sort of some epigenetic changes across these tissues. We have not done that yet. That is definitely on the docket for the next grant that is going to cover a lot of these downstream analysis. So now that all the monkeys have passed, we sort of have full access to all of the necropsy samples, plus all of the intermediate sampling. And I absolutely think that looking at some of the epigenetics and maybe making some comparisons to the methylation clocks that have been developed in a lot of other organisms would be super interesting, but we're just not there yet.
Don't worry, Tim, we're recording this. So all these ideas for future grants, we'll just send them along.
Can hold my feet to the fire on that. Huh? Okay.
So the last question is, you noted that lipid droplet sizes were increased in skeletal muscle of control animals. How do you know what is the process regulating lipid droplets in skeletal muscle?
So this is something we actually tried to confirm via RT-PCR because one of the reviewers asked about it, and looking at whether there were any changes in the plins, plin 1, 2, 3, 4, and 5, which would sort of control the accessibility of the lipid droplets. And we didn't see any big differences at the transcript level in any of those enzymes. So now, it's possible that at the protein level, they're very, very different. We haven't really done any proteomics on the muscle tissue yet. So as far as regulation of that, we don't know. All we have is just that we've observed that in the control animals, the droplets are bigger. In the CR animals, they're very, very tiny. And so we think that is indicative of a change in accessibility, but we couldn't see it by RT.
Great. Well, that sums up our questions, and I want to thank everybody who joined us today for a great talk on calorie restriction. I'm not sure the timing right before the Thanksgiving holidays was great timing, but we hope it inspires you for after the holidays. Thank you very much, and I hope everyone has a wonderful Thanksgiving. Take care.